So next, I will invite Dr. Ambrin Fatima to come and set the stage for us. She's going to talk about the rare disease landscape in Pakistan, because I think that is what sets the stage for the discussion that we will have throughout the conference today. Dr. Ambrin is an assistant professor here in the Department of Biologic and Biomedical Sciences at the Medical College, and also has a joint appointment at the Center for Regenerative Medicine. She is my partner in crime in the lab, so all the cool stuff that I see in clinic, she takes to the lab, and it's been a phenomenal partnership so far. So over to you, Ambrin.
Good afternoon, everyone, and thank you very much, Dr. Salman. So I'm very excited to tell you about the subject that I really love, and I hope at the end I can show you that there is actually much more when it comes to Pakistani genome that can show us the wonders and can help us in the precision medicine. So let's start with some, so this is the outline of my talk, and let's start with some numbers. So Pakistan is the fifth most populous country in the world with over 250 million individuals. But what make it really unique is the high rate of consanguinity when it comes to rare disorders. And the reported is more than 60%, but we know that there are some regions that have even as high as 80% of consanguineous marriages. The other factors are low literacy rate and the low spending on the healthcare system. But another thing that is very unique in Pakistan is like a lot of ethnic backgrounds and kind of a small founder type of a population that can also help us to decode the human genome. This is a global map based on the consanguinity, and if you look at Pakistan, it is highlighted as with the dark, and that increase the risk of rare disorder, especially autosomal recessive disorder. But even if we look at the other Muslim countries, those practice cousin marriages, so you can see the Pakistan is being on the top of this list as well. So because of the time limitation, I'm just going to show you one example of a rare disease that is primary microcephaly. Unfortunately, there are many misconceptions, and a lot of people do not even think that this is a genetic disease, but it is a purely genetic disease of cortex, where the children is born with a small head size and small brain. Highest incidence is in Pakistan, and there are more than 25 genes reported from primary microcephaly. And if we look at the number of genes, the red are the one where the genes was first time reported in Pakistan. So you can see an overwhelming majority was identified in Pakistani individuals. And on the other side, this pedigree is not a hypothetical pedigree. It was a pedigree of one family that we have identified. And this is very common that you see multiplex families where you have 10 or more individuals segregating with a rare disease. And of course, this increased the founder mutation, and there are many founder mutation. I wanted to put just one example, one founder mutation in ASPM gene, and that is very common in Pakhtun ethnic background, and we have seen hundreds of families with this founder variant. The thing that I wanted to highlight is that these cousin marriages bring the ancestral chromosomal parts together in a segment that we call autozygome or homozygosity, and that is very instrumental when it comes to mapping of the genome. Even before the NGS, we were able to map the genome through STR marker and then microarray, SNP, and now we are doing pretty much everything with exome or genome sequencing. And we know that this has been a wonderful tool to identify Mendelian genes. But what recently we have started seeing from the health and gene data and the UK Biobank data, that actually not only Mendelian, but consanguinity or homozygosity increase the risk of complex disorder like type 2 diabetes. And this is a wonderful study just recently published in Cell. So there's actually much more that we can even look at the population level. Another area that I wanted to highlight with reference to Pakistani population is the human knockouts. So since there is many homozygous mutation, and when there is a homozygous mutation, loss of function homozygous mutation that end up like knocking out the gene naturally from the human genome. And that, of course, if you look at the phenotypic spectrum, it starts from embryonic lethality with recurrent pregnancy loss, preterm birth, then sphere to mild Mendelian phenotypes, and go all the way to common phenotypes. But now we know, for example, from PCSK9 inhibitors and the trial those are going on, that there is much more in this area, and that can help us in therapeutic intervention as well. So one of the landmark paper that came in the field of knockouts, again came from Pakistani population. This was the first paper, and that actually suddenly got interest of all the pharmaceutical industry, and they started investing on this population and sequencing more individual. So on the recent data, they have sequenced 80,000 individuals, and they have found knockouts for the 5,000 genes from 80,000 individuals. And if you are wondering that if you want to achieve the same number from outbred population, you might have to sequence 11 million individuals. So again, this is a unique resource to look at the gene function. Unfortunately, we cannot really separate population genomics from rare disorders, because ultimately we have to go into the population. And we look at if the population data, for example, the latest version of NOMAD, the South Asian exomes are just 6% of the NOMAD, and the genome is only 3%. So this is, you can imagine the number of Pakistani genome and exome present in the NOMAD, and that's a big challenge when it comes to clinical genetic practice, for example, looking at the variant of uncertain significance. Again, another example I wanted to quote here is this missense variant in NOTCH3 that have been associated with the risk of stroke. If you look at this variant in South Asian, the allelic frequency of this variant is 1 in 98 South Asian. But if you look into European, so allelic frequency of the same variant is 1 in 2,600 individual. So again, this also kind of put the, that we should also focus on the population data and look at the allelic frequencies at the population level. Okay. Unfortunately, rare disorders or genomics have been at the lower priority, and because of the malnutrition, infectious diseases, we really do not have recognized the rate of mortality due to genetic disorder. There's no national newborn screening program. Genetic testing, if it's paid from the pocket, it is considered as luxury, and most of the population cannot afford it. And then because of lack of awareness, there are several social taboos and alternative explanation. For instance, like you might know about microcephaly, pure genomic literacy, not in the public, but overall, even the scientific community or medical community. So that's why there was not a lot of work on the rare disorders, but there are 3 models that we follow, or we work through these three models, and I'm going to these one by one. So one of the earliest models that have started on the rare disorder is this community research model. And these are basically non-clinical researchers going to the community, recruiting multiplex large families, doing the genome sequencing, identifying gene, going back to the family, reverse phenotyping, and these rely mostly on multiplex families. So we really haven't looked into the burden of de novo variant in this model. And this is how it really looks like. And they have, through this model, they have identified hundreds or thousands of genes, produced hundreds of PhD, and this is like, have worked well in non-clinical setting. But when it comes to clinical settings, so we have 2 models. So Dr. Chima is actually pioneer of this model. So it's a clinical research model where the clinician directly send their samples to diagnostic companies, international collaborator, get the clinical testing report. This was the initial setup of this model. And we know that there are wonderful findings that we have actually seen from this kind of research as well. Again, the model that we follow in AKU is a clinical genetics model, and where the clinician or the primary physician send their samples to clinical geneticist or genetic counselor, and then they mostly send to the diagnostic lab, get the clinical testing done, and there is pre and post-test genetic counseling associated with this model. If we look at the spectrum of disease, of course, it have adult cases as well as different disorders from the pediatric clinics. So previously, it was relying on mostly good phenotyping and selecting a good gene panel, but now everything is done through the whole exome sequencing. Okay, if we look at the clinical genetic capacity in AKU, so there was nothing before 2008, and now we have 2 clinical geneticists, we have 2 genetic counselors trained locally, and we have genetic nurses, 2 genetic nurses. So, but we still need a lot of capacity, knowing the high burden of rare disorder in our population. if we talk about the testing landscape, so for all the clinical testing or the genetic testing or exome sequencing is done through the commercial lab, but we do have an internal capacity for single gene disorder like beta thalassemia, SMA, or like other DMD or this kind of, or karyotyping. I also wanted to highlight some of the work. So I established my group in 2022. So my focus is on rare neurodevelopmental disorder, all the way from disease gene identification to disease modeling, and we use induced pluripotent in vitro-based model for this one. So we have recruited a number of families from different ethnicities, and this is how the distribution looked like. And we have around 20 to 30% novel genes, then we have like almost 20 to 25% variants. Those are already reported through the ACMG classification as pathogenic. And then we have a majority of variants, those are in the known gene, and that, of course, put a big challenge when it comes to VUSs. So we have established in-house pipeline to look at some of the VUSs where we think that they are pathogenic or likely pathogenic, and my lab is now working on several novel candidates gene and looking at the function of these ones. Another thing we have started building is iPSC resource, induced pluripotent stem cells. So we take biopsies from patient with the rare disorder as well as with the control, and then we go into reprogramming and characterizing induced pluripotent stem cell that can be used as a later for isogenic knockout and disease modeling. Okay, at the end, I would like to present what are the strengths, weakness, opportunities, and threat if we work on Pakistani population. As I mentioned, that this is a unique resource, and there is like a potential resource for knockout gene and also for protective variants, because we have large families, and they really help us to kind of recognize the variant. And we have large family size. We have a lot of early career genomic researcher who has PhD in genomics. And because of unavailability of resources, a lot of families are willing to participate in this kind of research. Unfortunately, the list of weaknesses is quite long, because we have only few healthcare providers when it comes to clinical side. There's no local infrastructure. There are cultural challenges when it comes to research. There are few funding opportunities, no clinical genomics program, and very important, there is no data on the population genomics. There is no data on complex genomics, complex disorder. And the work that we are doing is really fragmented, because even like there is no local collaboration. There's no sharing of data, because now we know that if you collect the sample or if you have sequenced the sample, that could be a wonderful resource to share with other researchers and can help us to diagnose diseases better. Of course, we can be part of a lot of DEI funding opportunities or collaborations. We can be part of clinical trial. We can start the Pakistan Genome Project, where we can sequence populations with different ethnic backgrounds. And we really need to start putting our effort to build biobanks and biorepositories, because I have seen hundreds of studies where people have collected samples, clinically diagnosed samples, but have used, those are really underutilized because they have been used just to look at a couple of single nucleotide polymorphism, and that is a wonderful resource that we can use for whole genome sequencing. And of course, we need to develop capacity in clinical genetics, genetic counseling, and we need to develop a lot more collaborations. Of course, overall, when it comes to translational research, there are only few funding opportunities in the basic science research, and it's really hard to compete with no preliminary data with international collaborations and international funding. There is healthcare inequities. There is large sequencing data coming for complex disorders. So even if we are able to sequence a couple of thousands, it probably would be difficult in that sense. And I think the one thing that we are not really aware of, like data utilization, that how we are giving the data to the pharmaceutical companies getting these reports, and we are not really, in future, if there will be any precision medicine initiative, we might not be able to help our population. So we really need to think in that area. And of course, there is no collaboration with pharmaceutical industry. And I think so one of the the most important is retention. And because of the current economic situation, we cannot retain our clinically trained people or PhDs, and they just try to find opportunities elsewhere. Okay, if we have to really work and bring genomics to LMIC, we have to work on different areas, and that is we have to build infrastructure in terms of labs, in terms of personnel capacity, computational resources, making the database, making the public database where we can actually look into the population-based specific variant. We have to work on educational and community engagement because, again, we have thousands of founder variants and that are increasing day by day because there is no awareness program, there is no clinical testing program, and there is no genetic counseling program. So we really need to work on a bigger level. What I have shown you is really just a couple of efforts, and it's really tip on the iceberg, and we have to work more, and there is much more that we can explore from this part of the world. And of course, ultimately, genome is our shared heritage. So that's what connects us. Even we are people of different color, different race, but that's where we are connected through our DNA. And I think so there need to be more inclusion of underrepresented population, and especially population like Pakistan, to have a better understanding of genome and bringing the precision medicine promise to the world. So thank you very much for that, and I would be happy to answer.